Today, we'll see about the introduction to mycology. So first, we will get familiarized with the terminologies which is used. What is mycose? Mycose means it is fungi. And mycosis is the disease which is caused by the fungi. Mycology is study of fungi. So fungi are generally, they are widely distributed in nature. They are present everywhere in the air, soil, and decaying organic matter. So they are eukaryote and they are heterotrophic organisms which are devoid of chlorophyll. They obtain its nutrient by absorption and reproduce by spores. Primary storage product is basically glycogen. So they are eukaryote. So the difference between prokaryote and eukaryote is prokaryote are the primitive cells. That is, they do not have any well-defined organelles, whereas eukaryote are organism which has well-defined organelles. So if you see, they have a proper cellular structures with nucleus, nuclear membrane, endoplasmic reticulum, mitochondria, and secretory apparatus. So they are facultative, anaerobe, and they are strictly aerobe. That is, they are strictly aerobe, and when there is even little amount of carbon dioxide present, they have better growth. So this is a cell of a fungi where you can see the cell organelles. So they secrete enzymes that degrade the organic matter into soluble nutrients, and they absorb passively or by active transport, and they are non-photosynthetic. So these fungal infections were recognized early as 17th century, and they were considered as a human pathogen. So trichophyton and Candida albicans were the common infections, and which was discovered almost in 19th century. And there are around 80,000 species which are described. Only 400 are species which causes infections in humans. So dermatophytes and candidiasis are the commonest disease. Dermatophytosis or oral candidiasis, if you see, dermatophytes are the skin infection where you can see the skin lesions, and which with their annular skin ring-like lesions, so which is commonly called as ringworm. Whereas these oral lesions, which are found, that is white patches, so seen in immunocompromised people, so it is oral candidiasis. So if you are coming, if this mycology field is almost less developed than the bacteriology field. So because they are not more frequent, they are less commoner than the bacterial infection. And if you see the mechanism of this fungal pathogenicity is very complex, and it is also very difficult for the humans to treat these fungal infection, since we need a longer course of treatment and longer durations of antifungals. So usually there is when will you see the increased incidence of these mycosis? So if you see this, these are the saprophytes. So saprophytes are organisms which depend on the dead and decaying for their nutrients. So they are usually opportunistic. So when you can expect this kind of infection? So when there is increased use of antibiotics or steroids or any immunosuppressive agents or immunomodulating agents. So these will suppress the immune mechanisms, and because of the suppression of the bone marrow, the cell counts will decrease, and it will pave way for more pathogens to enter. So if there is even the normal commensals or saprophytes which are present outside, they will also get they are more vulnerable for these patients. So who are those patients? Those who have immunosuppressions. If you see those patients who are treated for cancer or AIDS. That is, since HIV has been more common now, and it is the incidence is increasing. So HIV. So in case of organ transplants or even in stem cell transplantation. So in case of diabetes. So these are the condition which plays an important role in increasing the fungal infection in human. So what are the morphology of the fungi? So the morphology of the fungus, if you see, so there are two basic forms, that is yeast and mold forms. So what are those yeast? So yeast are usually unicellular fungi, so which are 3 to 15 micron meter in diameter, and it is usually spherical or ellipsoid in shape. How do they reproduce? They reproduce by budding. And sometimes these cells which reproduce by budding fail to detach from the mother cell, and they keep on extending. So it appears like a long chains of these budding cells which has not detached from the mother cell, which seems like a long hyphal filament. So they are also called as pseudo hyphae. So this is a one form of fungi, that is yeast. Yeast they are unicellular fungi. So this is a yeast cell where it reproduces by budding. So this is pseudohyphae where you can see the cells will not get detached from the mother cell, but they appear as long chains. So this is pseudohyphae. So next coming to the filamentous fungi. The next form of the fungi is filamentous fungi. So they are usually multicellular. So that was unicellular. This is multicellular filamentous colonies. And the hyphae is usually long thread-like tubular structures. So this entangled mass of the structure. So they are called as mycelia. Okay. So two forms are there. One is yeast form, will be the filamentous fungi, that is the hyphae. So that was unicellular, and this is multicellular. So this you can see the filamentous hyphae under microscopy, and this is a macroscopy on seen on any decaying organic matter, where you can see some fluffy structures. So they are the entangled mass of these hyphae, that is they are the mycelium. So coming to hyphae, so if you see the hyphae, it can be of various morphologies. It can be septate, or it can be a aseptate hyphae. So septate hyphae, so it allows free flow of cytoplasmic material. So other hyphae, the different morphologies, if you see, it is spiral hyphae, or it appears like a spring-like helical coils. Then you have this racket hyphae. So they are localized swellings by tight twisted hyphae, which resembles a tennis racket. Then we have this fabic chandelier. So they are numerous short branches appearing at the ends of the hyphae. So the basic forms here you can see the septate or aseptate, that is non-septate hyphae. So we'll see the picture of each. So this is a spiral hyphae. Okay, it's a spring like coiled structure, and this is racket. It's like a racket. This is favic chandelier where you can see like a branches coming from one end, which appears like a chandelier. So coming to the mycelium, so this mycelium, it can be a substrate mycelium or it can be aerial mycelium. So substrate mycelium, they usually in the level of the ground, that is in the surface, whereas aerial mycelium, they just arise from the surface where they grow. Okay, so this vegetative mycelium usually they have certain root-like structures which penetrate into the organic matter and they absorb all the nutrients, whereas this vegetative aerial mycelium, they what they do is they're basically they are aerial structures which bear the spores, which is responsible for the reproduction. Okay. So this is if you see the plate in usually you can see this aerial mycelium in the plate where you can see above the entangled mass of this mycelia you can see some structures popping out. So they are the aerial mycelium which bears the fruiting body which releases the spores which is responsible for the reproduction of the fungi. So now we'll come to the classification of fungi. So coming to the classification, we have morphological classification and based on the reproduction and based on the types of infections they cause, that is pathogenic classification. So based on the morphology, you can divide the fungi into yeast, yeast-like fungi, mold or filamentous fungi, and then it is dimorphic fungi. So what are yeast? So as already mentioned, so yeast are spherical ellipsoidal cells. They reproduce by budding. They produce creamy colored colonies. On morphologically, if you see the agar, you can see a creamy pasty colonies. And if you see the example for this is Cryptococcus neoformans. So you can see pasty agar, your growth over the media. Okay. And yeast-like fungi. So the only difference is the morphology of the yeast-like fungi is also same, where you can see the pasty colonies like your yeast. But the thing is, in microscopy, there will be presence of this pseudohyphae. So these reproduce by budding yeast, whereas yeast-like will have budding, which will not get detached from the mother cell, and so they will be appearing like an hyphae. So they are called as pseudo hyphae. Okay, and the yeast-like this Candida also is usually mostly oval, whereas your Cryptococcus is nice spherical structures. So this example for this is Candida. So Candida albicans is a species where it is one of the most common pathogenic yeast-like fungi. So coming to the mold. So mold or this filamentous fungi, they form the true mycelia. So they reproduce by the formation of spores. So they are the entangled mass of the hyphae. And one of the common example is your dermatophyte infection. Then we have this dimorphic fungi. So the dimorphic fungi is, so both yeast as well as this mold form will be seen in this dimorphic fungi. So when do they, in what forms, how do they they'll be seen? So if you see at 37 degrees Celsius, that is at a body temperature, they appear as yeast. Whereas at 22 degrees Celsius, they appear as molds. Okay, so this is called as dimorphic fungi. When you try to culture these organisms also, we have to take two tubes, where one tube you will keep at the room temperature and another tube will be in the incubator, where we will look for any dimorphic morphology of the fungi. So according to the morphological classification, we have four. That morphological classification: yeast like, mold, and dimorphic fungi. So coming to the fungal spores. So what are the fungal spores? So fungi usually reproduce by the production of spores. So spores are in general they are resistant to the adverse conditions, and they germinate only when the conditions are favorable. So they can be produced asexually or sexually. So asexual spores are mitotic progeny, and they are genetically they are identical. Okay. So they are of two types. One is conidiospores and the sporangiospores. So what is this conidia? So this conidia usually they are produced from this conidiogenous or some conidia forming cells, which is called as phyalid. So they are supported by a conidiophore. So you will see a conidiophore. On that you will be seeing a phyalid structure, and then you will be seeing this conidia. Okay. So what are the different types of conidia? You have microconidia and macroconidia. So what is this microconidia? So microconidia, they are small single-celled conidia, whereas macroconidia, they are multicellular structures. So here you can see the phyalids. So this is a aerial mycelium, which is a conidiophore. So from the conidiophore, you can see a broad structure, which is a phyalids. From the phyalids, you can see the long chains of conidia having the conidiospores. Okay. So here, in case of dermatophytes, you can see microconidia and macroconidia. So here you will not see any aerial mycelium like that, but you can see a single-celled tiny structures. So they are the microconidia, whereas like beans like long oval structures, which will have various septations having the macroconidia bearing the conidiospore. So this is a conidia. Next is sporangiospores. So sporangiospores, these are basically sac-like structures which bears these spores. Okay, so these sac-like structures, they are called as sporangium. So which is supported by the this sporangiophore. So there is a conidiophore, and here it is sporangiophore. So this is a sporangiophore where you will have a columella, and this broad part is apophysis, and that will bear a sac-like structure which is a sporangium, and the sporangium will contain sporangiospores. So based on the sexual spore formation, you can classify into zygomycetes, ascomycetes, basidiomycetes, or deuteromycetes. Okay. So mycota or zygomycetes. So they are the lower fungi with non-septate hyphae, and the sexual reproduction results in zygospore. So they also form asexual spores, which are called as sporangiospores within the sporangia. So they are the usually lower fungi. So the zygomycetes are lower fungi. So your examples will be Rhizopus, Mucor, Obsidium. So they are the zygomycetes. So this is a sporangiospore which gets the spores are getting released from the sac. Then we have this mycota ascomycetes. They form the sexual spores which is called as ascospores. So within a sac, this is a asci. So these are usually higher fungi having septate hyphae. They also form this exogenous asexual spores also. Sometimes they have asexual spores also, forming this in the they are called as conidia, or they will form sac also having the ascospores. So this is in case of basidiomyces and histoplasmosis. This is an asci. Then this basidiomycetes or basidiomycota, here sexual reproduction, it occurs in 4 progeny. So the basidiospores are usually supported by a club-shaped basidium. So they have a complex septa in case of basidiospores. That is in the common mushrooms and all, you can see the basidiospores which is seen underneath the the aerial structure. So this is a fruiting body where you can see the fruiting body underneath the fruiting body. You can see the basidium. Okay, the basidium. So each basidiospore, when it falls, what will happen is they undergo meiosis and then they form the sterigma and then the basidiospores are formed. And that will be releasing and it will arise, it will give rise to the newer fungi. Then we have another group of fungi which are the fungi imperfecti or deuteromycetes. So here it is a provisional group where the sexual phase has not been identified. So coming to the structure of a fungi. So these are the classification. Based on the pathogenesis, we can classify into cutaneous, subcutaneous, and opportunistic mycosis. So systemic and opportunistic. So cutaneous mycosis is the superficial layers of the skins are involved. Subcutaneous where the deeper layers are involved, and then we have this systemic infection where the organs are involved, and opportunistic where there is in case of immunocompromised conditions. So if coming to the structure of the fungi, we have a capsule which is present only in some fungi. Then that is in case of cryptococcus. Then cell wall, cell membrane, cytoplasm. So cytoplasm will have all the well-defined cell organelles. That is nucleus, nuclear membrane, nucleolus. Then we have this mitochondria and endoplasmic reticulum and other vacuoles will be seen. So in case of cryptococcus, we can see a capsule. So this capsule is usually a virulence factor. It is made up of polysaccharide. So it is seen in cryptococcus neoformans. So why the virulence factor is it is anti-phagocytic. That is, it cannot be phagocytized with the these macrophages. So this is one of the important virulence factor, and this is an opportunistic fungus, Cryptococcus neoformans. And you can demonstrate this capsule by the negative staining. So the cell wall of the fungi, if you see, they are usually multi-layered. They have polysaccharides, they have proteins and glycoproteins. So if the function is they provide shape, rigidity, strength and protection from the osmotic shock. And they also mediate attachment of the fungus to the host cell. So the cell wall polysaccharides, they elicit complement cascade and they provoke the inflammatory response. They are also immunodominant antigen which they elicit cellular responses and diagnostic antibodies. Okay, so the cell wall is one of the important component even for diagnosis as they provoke some of the antibodies which can be used in diagnosis of these pathogens. So cell membrane, if you see, they are bilayered phospholipids, and it is made up of sterols. So here you can see the sterols will be present. So they are ergosterol or zymosterol. So the functions of this cellular membrane is it protects the cell organelles and the cytoplasm, and they regulate the intake and secretion of the solutes, and they facilitate this capsule and cell wall synthesis. So based on the pathogenicity, I told you they are classified into superficial, deeper infection, and opportunistic infection. So superficial infection, if you see, they are usually mild and chronic infection, and they are caused by saprophytes, which has a capacity to digest the keratin. It includes tinea, ringworm, and common hair and nail infections. So deeper infections, it's also called as systemic mycosis, and they cause asymptomatic infection to fatal disease, mostly caused by soil saprophytes. Opportunistic infection, they are caused by avirulent fungi like mucor, Absidia, and penicillium, and they occur in patients with debilitating diseases like cancer and diabetes, where there is more immunocompromised conditions. So they are uptake of steroid and broad spectrum antibiotics. So now we'll talk about the lab diagnosis of the fungal infection. So what are the specimens which we receive in case of fungal infection? So the specimen which we receive are the tissue specimens, skin scrapings, then corneal scraping, blood, urine, CSF, and these are the routine specimens which we receive. So skin specimens which we receive, how do you look for? We usually, first thing is microscopy. So microscopy can be done with KOH or can be done with the KOH. So basically KOH we have 10%, 20% and 40% of KOH. So this 10% is usually, it's used for the skin scrapings, whereas 20% we use for tissue and 40% for the nail specimens. So when they send the skin scraping, they can send in a paper folded or in this in a slide. Okay, so they can directly do a KOH, put a KOH and put a cover slip and send to us so that we can look for any morphology. So why do you use KOH, potassium hydroxide? Why do you use? So basically we use this potassium hydroxide to digest all the keratins which is present in the cells, and this to expose the fungal elements so that we can be seeing these fungal elements very clearly. So this is a KOH of the fungal skin, where you can see these cells getting this digested, and the fungal filaments are seen. So here you cannot speciate, you can just tell whether fungal elements are there or not. Next, the stains which we use is calcopho. These are all from the direct specimens. How do we see? So the stains what we use is calcopher stain. So this calcopher binds to the cellulose, chitin, and fluoresces under UV light. Then we have this periodic acid shift and methanamine silver stain, or this fungal. These are the fungal stain where we do in the tissue sections. So this is a POA stain of the skin. This is calcofluor stain where fluorescence will be seen. Then once the fungus, the sample we get, we streak onto the agar. So if we streak onto the agar, we will look for the growth. So once it grows, we have to find what morphology of fungi it is. So what do? We have to take it and do the LPCB mount. So LPCB mount is lactophenol cotton blue mount. So here we just take bits of colonies, we just tease, that is we separate all these strands using a teasing needle, and we look for the microscopy. So what is this CB? That is lactose. It contains lactose, which preserves the morphology of the fungi. Then glycerol. So it prevents drying of the mount. Then phenol. It kills the fungi and it also acts as a disinfectant. Cotton blue. It stains the outer wall of the fungi. So this is LPCB. So this is LPCB of Aspergillus fumigatus, where you can see you can find out the what species it is. So what are the cultures which we use for the fungi? So basically we use this Sabrodes dextrose agar, Zepex medium, corn meal agar. So Sabrodes dextrose agar, it contains dextrose and some antibiotics like gentamicin and cyclophosphamide, because since very long term we are going to grow the fungi and it will be more vulnerable to these contaminant mold. So we also had cyclohexamide, and it is incubated at 37 degrees Celsius for days. So usually for slow growing fungi, it takes up to 2 to 3 weeks to grow, whereas the fast rapid growers will take only 2 to 3 days to grow, or it can be kept at room temperature or in a BOD, by that is biological oxygen demand incubator, where you can it will moisten the colonies, the fungal growth. It prevents the drying of the agar. So this is a Sabarod's dextrose agar, where this is a plate and this is a tube. You can see the fungal colonies. So this identification of the fungi is basically based on their colony morphology as well as your microscopy. So in colony morphology, we look for the color, the pigmentation. The pigmentation sometimes it can be seen. So we have obverse and reverse. Obverse is the front part of the tubes. Usually we do in a tube. So obverse is a front part and reverse is a reverse part of the tube, where you can see the pigmentation and also the rapidity of growth, how fast the fungi grows. And also we'll look for the microscopy. We'll tease it and look for the hyphae or conidia or fruiting structures. Based on that, we can tell what fungi it is. So yeast, if you see, they are usually on microscopy, as already said, they are oval or round structures, and they reproduce by budding or pseudohyphae. Whereas macroscopically, if you see, they are pasty colonies. It will be like a creamy colonies. It resembles like a bacterial colony. Mold, if you see, microscopically, you can see the hyphae, whereas macroscopically it will be cotton woolly or velvety. Some pigmentation can be seen in the reverse. Okay, so it will be a powdery velvety colony. If you look at the colony itself, you'll know it's a mold or not. So there are various antifungal chemotherapies which is given. So but it is often less developed because of the due to narrow spectrum, poor penetration, profound side effects due to extensive homology of the genes between human and host, and also increasing resistance to this antifungal drugs. So what are the common antifungal drugs we use? We use amphotericin B, flucytosine, azoles like ketoconazole, fluconazole, itraconazole. Then we have this echinocandins like capsofungin, micafungin. Then we have this grisofulvin, terbinafine, this clotrimazole, miconazole, nystatin, tolnafate. So these are the commonly used antifungals. So based on what type of infection, we can use the route of giving the antifungals also. If it's just a skin infection, then Topical antifungals like cotrimazole, terbinafine will be enough. Whereas if it is a systemic infection, then we have to take the orally or either IV route. And there are some uses of fungi also. They are essential in breaking down and recycling the organic matter. And they are very useful in food industries to make this cheese, bread, where fermentation is more common process is done. They are also useful for the source of many antibiotics like penicillin and immunosuppressants like the cyclosporins. They are, but these fungi are exploited by geneticists as models of investigation in case of many eukaryotic processes. Okay, so on the whole, if you see the fungi, we have classified the fungi based on the morphology as yeast and as well as the mold. And also we saw the morphological classification, the pathogenic classification and based on the sexual spores. And the investigation, what are the common investigation we do? So basically we rely on the microscopy and the fungal culture. So this is an overview of the fungus, of the mycology. Okay. So in the forthcoming classes, we'll be looking for the individual morphology of each of the fungi. Thank you.